Hey, good morning, everyone.
Good morning, sir.
Good morning. So let's
Good morning, sir.
let's start our class. Let them whoever wants to join, let them join. Actually, we are discussing about protein-protein interaction and what is the use of protein-protein interaction. Uh, is it be worth to study those type of stuffs because that is the next step or a big leap we are taking towards that because we are always looking at the translational part of whatever the research we are doing. That is lab to bed. That's the main thing. So we are planning, and whatever we are doing, it needs to reach the bed as quick as possible. That's what we are planning. We are trying all possible things, but it's having its own time and everything will take a lot more time. So that is the main interest of us. That's what if you look into a lot of research journals, currently a lot of people working on this pathways and network analysis. So even if you look into almost all the cases like COVID or any other viral infections, now they are talking about the network analysis. What are the proteins taking part and how it is going to be helpful to us. So the recent question regarding the pandemic is going on. Currently, they are talking about there is another variant, Delta variant they are talking about. So it is already there in our country. What is the efficacy of the vaccine we are using, that the Covishield and Covaxin, whether it will tackle that Delta variant? So that is the next important question. So how we are going to overcome this? So that is another thing. I am asking you the same question because whatever the vaccine, I already sent you the review article about the vaccine the first semester itself. So I hope everyone is having that article. Just go through that article and how they made the vaccine and what is the difference between the vaccines which are all commercially available. Just check it out and try to find out your own answer. And if you find time, we will discuss about that thing. So what is a variant? How the vaccine, whatever we are trying, is going to overcome this variant? That is the biggest question we are having now because we already lost a lot of lives in the second wave. In the first wave, too much restrictions and too much lockdown, so it was not that much hit in our country. But if you look into Western Hemisphere, US and Brazil lost a lot of lives. But in our country, the second wave, we lost a lot of life because the second wave, which came very rapidly, and we never prepared for that. So that is the biggest problem we had. So now we are talking about the third wave, and also we are having different variant. Variant means the same SARS-CoV. There may be some mutations will happen in certain proteins, which will be helpful for the survival of the virus. So we need to find how the virus is entering into the system and then how it is surviving. So what will happen if it stays too long? So already we saw a lot of problems in that. And another thing is in our country, we are not doing any post-mortem report or we are not doing any research related to the patient whoever died out of SARS-CoV-2, that is the COVID-19. But Italy is the only country who did a lot more research and did biopsy and everything from the patient who died out of COVID-19. They have taken all the samples and they have done a very good thorough study of those things. Even they have written a lot of articles. Now we are talking about ACE2 inhibitors, which is the main partner for the spike protein. The spike protein comes and sticking only to this ACE2, that angiotensin converting enzyme. From there only it starts propagating. A lot of researchers, they are interested to find the inhibitor for ACE2, But there are a lot of articles from the Italian researchers stating that it's not going to be helpful if you block ACE2 because they are having their own research output from the patient sample itself. So we need to thoroughly go through all these things. For that only we are having a lot more techniques. And in silico also we are studying all this protein-protein interaction because we need to find out a very good solution because it's not so easy to collect human samples. And that too in our country, it is not so easy. And in most of the Western countries also there are a lot of restrictions and guidelines to work with human samples. But if you look into the in silico works, what we are doing now, it may be helpful. So when you are taking some of the pathways and the network modeling, you can find out a lot of connectivity between the proteins, which are playing a very vital role. If you look into some of the virus or the viral proteins, they are not triggering the important proteins which are playing a very vital role in certain pathways. Instead of that, they are activating some other protein. By activating that some other protein, it's malfunctioning the whole system itself. That's what happening in the case of dengue and in the case of SARS COVID-19 also. So once if you look into the sampling or the patient cases, they are triggering something, so they are ending up with a cytokine storm in the case of COVID-19. And if you look into dengue, it's leading to dengue hemorrhage. Dengue hemorrhage is happening because there are a lot of malfunctioning happening with the pathways, which is not, the virus is not directly having any connection to those things. If you look into the complement pathway itself, they are smart and they are activating or escaping the system by activating some other protein. So it's masking the immune system so that the immune system can't recognize this as a foreign pathogen. Instead of recognizing this as a foreign pathogen, it thinks this viral protein as a part of our own system. Instead of killing those things, they are helping those viral proteins. So that's what's happening in the case of dengue and COVID-19. So we need to thoroughly study the mechanism of virus, how it is entering, and how it is using the host system. Without thoroughly analyzing all those things, if we are designing something and if we are coming up with some result, then it's going to be lethal. So that is the main thing we are going to study here. So let's get into the PPT. I think the PPT is visible. You are able to see the PPT? Hello?
Yes, sir.
Yes, okay, thank you. So if you are looking into this protein-protein interaction, so what it is going to do, that's one of the important things what we are always targeting is cancer biology, because cancer biology is somewhat complicated. So only at the final stage we are coming to know about these things. So if you look into this protein-protein or protein-protein interaction, we will come to know that how to suppress the tumor. So instead of suppressing the whole system, whatever the treatment now we are having is, it's also affecting the normal cells. So we need to find out a solution to target only the cancer cell so that it will not propagate. So that thinking that in our mind, we are doing all these things. And then we already discussed about pharmacogenetics. That is very important in the case of drug transporters and drug receptors and some of the important drug targets. That's what we discussed in the beginning itself, the pharmacogenomics and pharmacogenetics. So if we know what type of mutations they are having, then only we can address the question more strongly. So there are a lot of protein-protein interaction, whatever you know, and it's very big. So these are called as scaffolding proteins. Scaffolding proteins plays a very vital role in. If you look at the scaffolding protein, here we are talking more about the kinase pathways. These kinases are playing a very vital role And if you look at the most of the kinase, they are the targets. Whatever the disease you are taking, especially in the case of cancer, these kinases are the main target. So if you look into the chronic myeloid leukemia, the TCR-ABL tyrosine kinase is one of the important kinase. So whatever the drugs which are commercially available are targeted towards that. So like that, in each and every disease condition, we need to find out. And then another protein-protein interaction network is metabolic pathway. So metabolic and signaling pathway plays a very vital role because if you are taking something, it needs to get metabolized. If it is not get metabolized, if it is accumulated, and we will come up with a lot more problematic things. So that's what, if you look into the steroids, whatever we are taking, certain steroids, we can't metabolize immediately. So there is a time period for everything. If you are taking some steroids, means maximum they will allow you to take it for a month, and it starts from 5 days to 30 days, and then the doctor will advise not to take more than that. So the steroids gets accumulated. So there is another question. You know, most of the bodybuilders, so everyone knows about the muscle guys. What other? You don't know any muscle guys? If you look into the bodybuilders, how come they are getting the muscles like that? Do you have any idea? So most of them are induced by steroids. And then in certain games also, they will take those steroids, and then only they can hit. If you look into baseball, they need to hit home runs. Home runs are not so easy. So some of the guys like Barry Bond, not James Bond, his name is Barry Bond. Barry Bond is one of the guy in US who was the most valuable player who hit a lot of home runs. So the ball needs to get out of the stadium itself, out of the playground itself. Then only they call it as home runs. So for that they need a lot of energy. In some of the games, they are allowing these steroids. So I think in tennis also they are allowing it, uh, but not in Olympic or any other athletic. They are not allowed to take this type of steroids. So there are a lot of restrictions are there. So these pathways, these metabolic pathways, are playing a very vital role. So if something is happening, then we can't do anything. We need to find out a solution to stop or to fix this. So if you look into the morphogenic pathways, there are a group of proteins which take part in the cellular function, especially during the stage of developmental process. During the stage of developmental process, there are a lot more proteins. They will come and they will do a lot more work in the developmental stage, because that is the stage where the thing fetus needs to develop very fast. And then comes the structural complexes and molecular machines, which are having a lot of macromolecules, especially the proteins, which are brought together. So even if you look into the complexes, whatever the complex we are studying about during the production or during this glycolysis, so there are a lot of complexes involving in the complex 4. It's a very big protein complex. So even if there is some malfunction is happening in any one of the protein, then it will not function properly. So that's what I already told you. If there is some mitochondrial DNA mutation means, then there will be a problem in the mitochondrial complex. So especially in the case of this eye, there are a lot of complexes involving in this functioning of this messages carrying. That's what they call it as axons. So the axons play a very vital role. So if there is some mutation is happening in any one of the complex, the messages will not transfer properly. So there is a lot of research which are going on that thing. But so they already reported what are the different types of mutations. Due to the mutations, the complexes are not able to make the necessary protons for that. The protons are not made means the ATP synthesis will not be there properly. So whatever the energy they need, it will not be supplied properly. If the energy is not supplied properly, then the messages will not carried out by these neurons. So that is the biggest problem we are facing now. So certain cases, the eye, they call it as Leber hereditary optical neuropathy. So the eye will be right and the brain will be right, but there will be some problem in the message carrying nerves. So, but it is not the case with some of the organisms like frog or otherwise goldfish. If you destroy or cut the pathfinding nerves, they will regenerate. But in our cases, regeneration will not take place. So that is another problem. Leber hereditary optical neuropathy occurs in a lot more cases. If they are consuming too much of alcohol, because now whatever the alcohol they are consuming, it is not 90% or 99% ethanol itself. There is some concentration of methanol is also there. Methanol is prone for this type of things. It will completely affect the nerve system, especially in the brain and in the pathfinding nerves. So there are a lot of studies which are going on. So this is one or two examples related to this. So if you look into the signaling pathways, that's what I already told you. Based on the signal, they need to get the essential things. That's what I already told you about that Parkinson's. For Parkinson's, they will give you L-DOPA. So dopamine receptors needs to get the signal properly, and based on that, they need to get the DOPA, L-DOPA. Then only the brain will function properly. Otherwise, the brain cells will get damaged and uncontrolled movement will take place in the body. It will not have any coordination. The leg will move one side and the hand will move on another side. It's not so easy to control it. So these are some of the things we need to address. So these are some of the signaling pathways which are playing a very vital role. And then if you look into morphogenic pathways is very important in the case. There are a lot of proteins involving in that in order to do some function. So one single function, a lot of proteins comes together and they did the job. If there is some malfunction is happening there, then finally it will lead to a lot of other problems. So another thing is we are having a very good mechanism. Once the age is picking up, that's what we call it as the biological clock. The biological clocks are pretty good. They know the system very well. So that's what we call it as age-related diseases, because the biological clock is having a time period, and finally, whenever the time comes, it will shut down by itself. So these are some of the pathways, and then the other thing is structural complex. That's what I already told you in the first one, that complex. Fourth, everything plays a very vital role. Here we are talking about chaperones. Chaperone and chaperoning are playing a very vital role. So they will be there at the time of protein folding. So it will assist the protein to properly fold. Once the function is done, then the chaperone will not be there. So it will destroy itself. It's having a very limited time period. So if the chaperone is not there, means then the refolding process will not take place. That's what they are telling or claiming, that chaperone is also playing a very vital role in the case of Alzheimer's, because Alzheimer's is mainly caused because of beta amyloid formation or plaque formation, which got deposits in the cells in the brain. Once it gets deposited over the cells or it accumulates, then the cell will lose its function. So that's another thing. So we need to harness how to get rid of those proteins that have beta amyloid formation or plaque formation, to get rid of those things. So there are a lot of things which are involving in that. So how to find out the protein-protein interactions? So there are a lot of methods we are having. So one of the things is we need to have some fusion proteins so that it will go and bind to the protein which are having very good relationship, or the interaction will be good with certain proteins, because all the proteins will not interact with all the other proteins. Only certain proteins or a group of proteins will have its own functional partners. So that if you fuse it with a tag, and we can remove that protein from the thing, and then we can either use MALDI to find out the sequence, because we know whatever the protein we fused or tagged with that, and then we need to know only the partner protein. So that protein we can purify it, and we can analyze it by running it on a 2D gel, and then take it out, and finally do that MALDI, and we can find out the sequence, what are the proteins are present there. And then co-immunoprecipitation. So that is the other important things. If you have any particular antibody to that, you can bind to that, and then collect the protein out, and then take your antibody out of that, and then find out what the protein is interacting with this particular thing. That's what we call it as co-immunoprecipitation. And then these two hybrid is a pretty good technique, whichever is, because we are having the yeast to 2 hybrid system, which is already standardized, and then you can put whatever the protein of interest over there, and you can elute your thing and find out what are the things present in that. And then comes the Diacor. Diacor is a direct step. That's what I already told you. It's a very good instrument where if you know both the proteins, you can find out the interaction. So how strong the affinity between the two proteins, whether it is having a weak interaction or a strong interaction, that type of things. And AI from atomic force microscopy is now only it's picking up. The problem is just it will give you in a different way, what you call it as the interpretation is not that much easier. Now only it's the field is picking up, it's slowly coming up. So it will take some more time to get it out. And then fluorescent resonance energy FRET analysis. FRET analysis is also a solution study. So this study is also they are working on that. It's not so easy. That's what there are a lot of groups working on that, and the technique they are also trying to improve it. Once it comes out, then it will be pretty good. Still, there are a lot of works are going with FRET also. Another one is the X-ray diffraction. It's one of the standardized method where if you get the crystals, which is not an easy process, then you can find out how the protein is interacting with the binding partner, and what are the amino acids are also involved in this. That's very important thing with the X-ray diffraction, because it is one of the foolproof. That means 100% whatever it is having, it will give you the exact result. So it's a pretty good technique. But now the technique is pretty good, but getting the crystals are not so easy. So the rest of the things it's advanced. Like anything else, collecting the data using the synchrotron radiation is pretty good, and then solving the structure is also pretty good. But the problem is getting the protein and also getting the complex crystal is not so easy. So now another thing is what we call it as cryo-electron microscopy, where the technique now it's improving like anything else. So that's a pretty good technique. So this is the right time to start going through all these things. That's what experimental methods, whatever I already told you. So that is the same thing. And then comes that bioassays are there, where you can do that pull down assay and find out what are the proteins are involving in this and how they are interacting with this. So this is the thing about protein interaction and everything. And now we are going to look into the databases. So build is one of the old database, what you call it as biomolecular interaction network database is there. And then it's having a lot more data. And then comes what types of algorithm they are using, and then how to use bind. So I already asked you people to work out with some of the network tools. I don't know how many of you started with that. I told you eCell or vCell or Cytoscape. So you can try with glycolysis, which is a simpler pathway, where you can find out how the things are working. I don't know how many of you tried those things. Have you ever tried? Have you ever tried any of these tools? Yeah, Maria Jose has sent a question. Neurofilling. It's entering through neurofilling. I don't know. That's what each and every day they are talking about new targets for SARS-CoV-2, but we are not sure. So if neuropilin is a good target, I'm not sure about that. But we need to find out what is the role of neuropilin. So daily, there are a lot of works are coming up, and they are trying to find it out. Even vaccines also in our country, each and every state government is putting pressure to finish the first phase of vaccination, which is not a easier job. If neuropilin is a good target, it's fine. And then recently they identified that from the beginning they are telling it's man-made virus, that SARS-CoV-2. It started in China and it is made by some of the virology lab in China. And US already identified that thing. And then they already sacked the NIH director. In the beginning, he was supporting China that it is not a man-made virus, SARS-CoV-2. But now there are a lot of US employed a lot of people to find out whether it is man-made virus or it is an accidental thing which came out of the lab. And now they identified this completely made by some of the Chinese group, and they purposely released it out. That's what they are telling. Based on that evidence only, they sacked the NIH director Fauci. Fauci's name is Fauci, and then they sacked him. So that is the recent news. But anyhow, we need to find out a drug candidate, potential drug candidate. So whatever Maria Jose has sent here, I'm not sure about it, and I'm not sure about what is the function of neuropilin, how it is interacting with the SARS-CoV-2. I'm not sure about it. I need to update it. If I come to know about the thing, then I will explain it to you. So, any other question? No, each and every day they are telling a lot more new targets. But how far it is going to work, no one knows. Other thing is, if you are increasing our immune system, that system will take care of this thing, foreign pathogens. That's what we need to find it out. We need to have our own antibody for that. So that is the only thing. If we find it out, then it will be a very good thing. So we need to find out. Any other question from your side? Yes, other.
No, sir, no questions.
No, okay. Anyone, any questions means we'll discuss. Because how the pathways plays a very vital role, very interesting. And I'm not sure if you find time, just I hope if we are reopening, that would be nice, so that if you come here, we can do a lot more work on pathway analysis and everything. Okay.
Is it chance after the semester exam?
Huh?
Is there any chance to opening the university after semester exam?
Yeah, first for that they need to vaccinate each and everyone.
Most of the people we have vaccinated.
then it will be good. If everyone gets vaccinated, means then the government will open everything. Because that's what happening in almost all the European countries and in US. Now everything is normal there because everyone got vaccinated, and almost all the educational institute, the Europe, everyone is going to work. In our country, now only they started that vaccination formalities. So they need to make it a must, so that if everyone got vaccinated, means then we will start, everything will run up. Otherwise, it's a big loss to us. I'm not sure whether I'm reaching you or not. So that is another big problem. Online, I'm doing something. Whether it is reaching the student or not, that is another big problem. And application side also, if you are here, means then we will go ahead and do everything in the lab. The discussion will be very good. Now you are in a remote place. I don't know whether you are having the necessary things to access some of the whatever the tools I'm telling. I don't know whether you are able to access or not. There are a lot of practical difficulties we are facing. That thing we can remove it once you are here. So we are hoping for the best. I hope everything will be all right. So because another thing is vaccination for a country like us is not an easier job. 130 crores of people, and then that too they need to get the second dose. Calculate it. Is 130 plus 130, 260 crores vaccine we need to get. So which is not an easier task. So when you compare with the other country which is behind us is US. US is not having this much population. They are having only 50 crores, 50. At the most, if you add plus 10 crores also, it is coming around 60.
Sir, no, sir. Actually, US has very less population than us. They have only 32 crores or 320 million.
No, they are telling that, but they are around 50. But I'm not sure about that. Which year you got the data, I'm not sure, but they are next to us. Next population wise is US. Even if you put 30 plus another 10 crores also, it will come around 40 crores also. So compare the population difference. It's nearly 100 crores we are having excess. So we need to make vaccine, and then we need to make available for each and everyone. Another thing is it's not an easier job. That's what I want to make it. You need to produce that much vaccine within a short span of time, because we are expecting the third wave at any time. So in the meantime, we want to vaccinate almost how much is possible. They are doing it. I'm not sure. In Tamil Nadu, it is very fast. I don't know whether you got vaccinated or not. Most of you need to get the vaccine. Anirudh, you got vaccinated? Huh?
Yes, sir. I got vaccinated a month ago.
Oh, then it is good. I hope all of you get the vaccine. And now it is, they are calling, it's not necessary to go for registration or anything. Once you reach the center, you can.
Yes, sir. They're keeping a lot of drive-through vaccinations camps or walk-in vaccination camps. You can just go
That's
and.
what I'm telling. Especially
Yes,
only in Tamil Nadu it is happening. In most of the northern states, it's not happening. Tamil Nadu, they are doing it. In each and every small town, also they are having 3 or 4 vaccination centers, camp like thing is going on, and they are doing it. But we need to look into other states also. Some of your seniors who are from Kerala, they told they are running out of vaccine. They are not having enough vaccine. That's what I heard from your seniors. But I am not sure. the neighboring states itself is having some problem. So we need to get the vaccine and vaccinated. That is very important. Everyone needs to get the vaccine. So then only we can stop the next wave. Okay. So if there is no question, we will break at this point. We will continue tomorrow. Okay. So computational biology students. Hello.
Yes, sir.
Yes, Satish, you are the one replying to me. I'm not sure about others.
Yes, sir.
Yes, sir. Yes, sir. We are.
Yes, sir.
I don't know whether you all received that mathematics notes, whatever I sent.
Yes, sir. We
Yes,
received
sir.
it.
We have received. Sir.
Just to go through the problems, both worked out problems and also the exercise I have given there. And if you have any question, we will discuss it tomorrow, and we will have a internal based on it. Okay. So just go through the thing. Okay, I got a reply only from one student. The rest of them I never received any reply. So just I want to make sure whether you receive it or not. Okay, thank you. Yeah, there is no question. We'll break at this point and we will meet tomorrow. Thank you.
Thank you, sir.
Thank you. Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.